Hello. I'd like to give you a little insight into some possible things you can do in labs that may not be as cost prohibitive as things in the past. And part of this came out of a program I was in the summer of 2016. It's an RET program through KU. And one of our, we were charged with the task of trying to create at least a lesson plan slash lab that we could actually introduce to other teachers that could possibly help them with various lab skills, for instance, in their classroom. So what we came up with was very simple, a do-it-yourself student colorimeter. And you're probably saying to yourself, well, we've got colorimeters from different companies. We can buy them. We can have them in the lab already. That is a true statement. But what we are offering is completely different. It's going to be a very, very simplified version of a colorimeter. And the neat thing is, students put it together, and students have to troubleshoot any problems that go along with it versus you having to look at a piece of equipment and see if it's hooked up right. So why do we want to do something like this? Well, we want to teach lab skills. We all need lab skills. Students need lab skills, and they're lacking a lot of different skills. Problem-solving skills, which is part of the problem, a lot of times they find when they just push a button on a machine and it reads the number, they type the number down and they don't have any idea what the number means. They're just collecting data, so to speak. Well, they'll be collecting real data with this device we've got here. And the other aspect here, which is the neat part of it, is it allows for student design projects and labs. Let's have students design a lab and collect some data and try to interpret what they get. That's really the ultimate goal here, hopefully. So most of you might have something like this in your class, in your lab, a Spec 20, okay? We've got one or two of these at our school. If we've got a lab intensive activity where we have to read out absorbances or transmission, it's one student at a time with this, especially if they have multiple trials to go through. It's a very slow process. It takes days to get the whole class through something like this, okay? And that's why a lot of teachers sometimes will just use this as a demo. Well, that demo by itself, all the students look at this and they think, that's just a big black box. I know I put the tube in there, and I look at the number here, and I write it down, and I'm ready to go. Well, we want the students to try to get a little more out of it than that. So this is cost prohibitive in that context. The other options you've got now are there's some much cheaper options. A vernier colorimeter. A lot of times they have 3 bands at 3 specific wavelengths you're looking at. And they're very good devices, and they're very used quite a bit more now than I think Spec 20s. But again, they're about $250 each. What we would like to introduce to you is something much, much more student-friendly. So a multimeter you can get at any Radio Shack and or Harbor Freight. We got ours for this activity for $6 each, okay? Standard cuvettes, if you've got those in your classroom for another device, they're the same size, so they're standard size. We got these, about 100 of them, for less than $15 on the internet. So that $15 goes a long way on those. LEDs were just devices, little assorted LED packs you can get at like a Radio Shack. These were less than $4 for a assorted pack of 20. We also purchased a CDS photocell, which it was a pack of 5 of these for about $4. Okay, and then we also used for the source of energy here was a 3-volt CR2032 battery, very common battery you can find at stores. Where we've changed the game here is we've added this 3D printed colorimeter. It's a 2-piece colorimeter, and it has a cuvette cover. Okay, and we'll take a look at what this is. Though everything on this page, when you combine all this together, it is way less than $20 for this setup. And what's neat about it is this is pennies to actually print these if you've got a 3D printer, and we have all those files for you. So what would you, first of all, if you hand a kid a multimeter nowadays, they don't know what that does, what it is. It's got a lot of dials on it, etc. So one activity we start with is, if you're going to use this equipment, you got to know what it does and how it works. So one introduction to doing that is very simply getting a note card, having them write with a pencil, a graphite line, pretty thick, and then you're going to have them try to determine the resistance in ohms at different distances along this line. A very quick data set we collected real quickly from 5 different centimeters, and those are the resistances we got. We graph that. We got a line of best fit. We get a 0.99 correlation coefficient, which means that's a pretty good idea. It gives you a very good idea what this And at least it does to me. Now, to a student that does this, they need to look at this and try to determine what is the relationship. Is the resistance going up as I go away from my point of origin, or is it going down? Again, problem-solving skills using graphing, and also probably introducing some Excel spreadsheet activity if you could. Now, the meat of what we've got here for our device is this. We've got this, and this is just looking at it from the top off. This is the base plate of the 3D printed colorimeter. We've got a 3-volt battery that essentially powers the LED, many different LED colors you want. This is the opening for the cuvette to be sitting in. And then this is the CDS photocell that receives any light that comes through the liquid. So based on the liquid you put in here, a certain percentage of that light will either come through or will not come through. And based on that, this is going to read a certain resistance. These are connected to your multimeter leads. All right. So what happens is the cuvette is essentially housing the liquid that you're trying to determine. Okay. A concentration of. So this is a very simple operation. The only difference here is if you're actually going to look for absorbance, all you do is you take the log 10 of your resistance value, and that will give you the absorbance, and that's based on a common formula. So the benefit here is you've got any number of different LEDs you can use. The question is, which LED do you want to use if the liquid's red, or if the liquid's blue, or if it's green? What's the best LED to use? Those are questions you could answer, but it would be much better for a student to determine which one's best to use. And how would they do that? Well, they'd have to set this up, set up some experiments to determine that. That way they actually have a very good working knowledge of the whole device. Okay? So the setup really looks something very simple like this. You're going to have a cuvette here that you pull the cap off, the cover off the cuvette, you put the cuvette in, you put the cap on. It's pretty straightforward. Making sure that you're reading ohms at the right level according to the instructions. And then this is really an example. So you could take any colored liquid. In this case, it could be red food coloring. And I serially diluted them. So I've got these dilutions, and then I put each one of these in the do-it-yourself colorimeter, recorded the ohms that we're reading, okay? So we recorded the ohms, okay? And then after that, I took the log 10 of each one of these resistances. So that's what the log 10 of each one of those are. So I graphed the concentration versus the log 10 resistance. And this is when we used a red LED. We did the same thing for a yellow LED, and we got a different graph that's fairly close to this one. Okay, just to show you that variance is based on what equipment you're using. So after getting this done, you can use this calibration curve that you produced, and this one was produced in Excel. Very simple. I got a 0.98 correlation coefficient, got a line of best fit. You could determine the absorbance of an unknown, get the log 10 of that. Once you find that out, you can go across, hit this line, and you can estimate the concentration of in this case, it was copper sulfate. So the data from this data table and this graph are for copper sulfate, which is a common blue liquid used in chemistry. Okay, and it's the value of this is being able to having students determine concentrations of unknowns. Okay, so the setup's fairly simple. These are fairly cheap pieces of equipment you have to get. Once you get them, they last for a long period of time. The watch batteries will last for 12 hours straight if they're on full time. Intermittent throughout the classroom, they'll probably last for 6 to 10 days based on some estimated work we've done with them. The actual, in this case, we printed these, not the cuvette, but the top and the bottom of the colorimeter and the cover on a 3D printer. The printing did not take that much. It might have been 20 minutes to print all three of these things together. Again, if you have access to a 3D printer, this is a very simple thing to do. We've provided you all the STL files you'll need to print those. So you'll load the STL files into your 3D printer, however you do that, and then it's ready to go. So when can a student use this outside of just determining the concentration of red food coloring? Well, If in biology, a lot of times we use different solutions, and these are all colored solutions that have color variance based on how much of what we're looking at, the target molecule is. Glucose, starch, proteins, and vitamin C. These are solutions that will change color based on how much of that is present. So it's possible a student could try to determine how much glucose is in something using a Benedict's solution, cereal dilution. Okay, so these are very common in biology, but what is really neat about the whole setup here we've got that makes this very beneficial to you is students can come up with their own ideas. One possibility is you can actually throw out some open-ended questions like how much chlorophyll is in this maple leaf versus this dogwood leaf. Is there a difference? Is there a difference in concentration of chlorophyll? It could be any pigment that you might be able to extract from something. Okay, and again, any of those are possible ways you can quantify it using this colorimeter. But we can come up with some very quick ideas for students. But when they come up with their own ideas, that's even better, because then they're actually becoming real scientists. All right, so it's very simple. Would you like to have something that's very simple, cheap, inexpensive, student-oriented, and student-driven and student-understood? Or would you want this big black box that costs about $2,000? And they're still very usable pieces of equipment. Our suggestion is very simple. When students can put it together, they can fix it if anything goes wrong with it. And they also can troubleshoot it when something goes wrong here. Students really can't troubleshoot this. It's pretty much up to the teacher to find out how to fix this, or call somebody else who knows how to fix it. If you want any of the files, or if you're interested in any of the lessons for any of this, if you just email me at wwelch@usd260.com, I'll send you all these files, and hope you enjoyed it. Thank you.